chromosome and DNA mutations. There are 4 kinds of chromosomal level mutations. There is duplication, a segment of chromosome that has been doubled; deletion, a segment of chromosome is deleted; inversion, a segment of the chromosome is turned 180 degrees; and translocation. A segment of chromosome moves from one chromosome to a non-homologous chromosome or to another place on the same chromosome. There are 4 categories of nucleotide mutations. The first are single nucleotide substitutions, which is one base pair change, and there are 2 kinds of single nucleotide substitutions. Polymorphisms, which are single DNA base pair changes found in more than 1% of the population, and mutations, single DNA base pair changes found in less than 1% of the population. The second kind of nucleotide mutations are deletion-insertion polymorphisms, or DIPs. They are short insertions or deletions of a single or a few base pairs. The third is simple sequence repeats, or SSRs, which are 1 to 10 base sequences repeated 15 to 100 times in tandem. The last mutation is copy number variants, or CNVs, which are large blocks of duplication or deletion with a population frequency of less than 1%. There are multiple kinds of single nucleotide substitutions. The first is polymorphisms, which are single DNA base changes found in more than 1% of the population. The second is mutation, single DNA base changes found in less than 1% of the population. The third is nature of base substitution, which has 2 subcategories: transition, which is the substitution of a base of the same type, and transversion, a substitution of a base of the opposite type. Under mutations, there are 3 subcategories: missense, which alters a codon so that it specifies a different amino acid in a protein; nonsense, which converts an amino acid specifying codon into a stop codon; and a silent mutation, which alters a codon into one that codes for the same amino acid. There are 2 kinds of deletion-insertion polymorphisms: deletion, which changes the number of DNA bases by removing a piece of DNA that can be a single or a few base pairs long; and insertion, which changes the number of DNA bases in a gene by adding a piece of DNA that can be a single or a few base pairs long. The mutation called simple sequence repeats, or SSRs, are repetitions of one or more nucleotides directly adjacent to each other. The last mutation is copy number variants, or CNVs. When there is unequal crossing over, it produces new alleles of CNVs. There are large blocks of duplication or deletion with a population frequency of less than 1%.